Pharmacogenetics is an emerging science that helps explain the reactions people have to various medications, based on their genetic makeup. This is a lot to take in, so let's picture this. You and your two friends are going shopping. You all have a very similar style and are in the market for a new dress for your upcoming graduation. You decide to talk to a sales consultant who assures you that she has the perfect dress for the occasion and puts you all in the same dress. There's just one problem. The dress fits your one friend perfectly, while it's too long on you and too short on your other friend. You all come out of the dressing room and inform the stylist of the problem, but she informs you that the dress is the only one that fits for the occasion, and that the store takes on a one-size-fits-all approach. Now, picture this in a medical setting. You and several others receive the same diagnosis and prescription. The result is the same as the shopping example. In this one-size-fits-all approach, the drug will only be effective in a small amount of the population. But why is this? The answer lies in our DNA, specifically in our genes. No, not those genes. Every person has their own set of unique instructions that helps make you, well, you. This is called DNA. DNA is packaged into chromosomes, and half your chromosomes come from your mother, and the other half come from your father. Each chromosome contains sections of DNA called genes, and some of these genes help code for specific traits. Today we'll be focusing on genes that encode for enzymes. Enzymes are a protein that helps make a biological task happen faster. Enzymes involved in the cytochrome P450 family break down different drugs. Mutations within the genes that encode for these enzymes affect how well the enzyme will work and disrupt the way that the enzyme metabolizes the drug. Because you inherited a set of instructions from both your mom and your dad, there will be two types of enzymes that are produced. You may have a combination of functional enzymes, dysfunctional enzymes, or some combination of both. The combination will directly impact your ability to digest drugs. In order to determine what genes you inherited, doctors can order genetic testing to look at the known gene locations to determine if there are any changes to the DNA that might be affecting your drug metabolism.
Let's use the drug warfarin, which is a blood thinner, to see how genetic testing contributes to improved patient outcomes. Warfarin is a drug that causes more trips to the ER than any other drug among older adults. In your body, vitamin K promotes blood clotting proteins that allow blood and platelet cells to stick together when an artery is clogged with plaque or to stop bleeding. If you are at risk for blood clots, warfarin can be used to inhibit your body's ability to make vitamin K, and therefore the ability of blood clots to form. It is important to have the correct dosage of warfarin. If too little is prescribed, blood clots may still be a risk, and if too much is prescribed, there is a risk of excessive bleeding and overdose. There are known mutations in 3 different enzymes that contribute to warfarin dosing. One enzyme helps create the active form of vitamin K. Another is responsible for vitamin K availability in the body, and the third enzyme breaks down warfarin once it's in the liver. If a patient has a mutation that causes the inhibition of vitamin K production, then giving a standard dose of warfarin to that patient would cause vitamin K production to drop to dangerous levels, which could cause excessive bleeding. This is an example of a mutation that results in slow metabolism, as there is not enough functional enzyme to break down the drug, so the drug stays in its active form for a longer period of time. This patient needs a lower dosage of warfarin to achieve the wanted response. On the flip side, if a patient has a mutation that causes vitamin K to accumulate in the body, giving a standard dose of warfarin would not be enough to appropriately reduce vitamin K levels. This is an example of a rapid metabolizer in which the drug is used up too quickly and will not have an effect on preventing blood clots. This patient would require a higher dose of warfarin to inhibit the clotting pathway. Additionally, if a mutation occurred that inhibited the enzyme responsible for breaking down warfarin, this patient would require a lower dose of warfarin in order to avoid a toxic buildup of the drug in their body. As you can see, people metabolize drugs at different rates, and the correct drug and dosage combo for each person is essential for the best patient outcome. Genetic testing would allow providers in any of these cases to make adjustments to the dosing of warfarin, and this practice could be extended to other drugs when more research has been done. Warfarin provides a somewhat easy guide for how dosing may need to be changed because of the known genetic mutations associated with enzymes specific to the metabolism of this drug. Although this is not the case for many drugs, current research about drug metabolism in each individual is slow and more difficult to complete than standard medical research. This is part of the trade-off needed to improve personalized medicine, such as in tailoring drug prescriptions to individuals. Scientists know that the 10 highest grossing drugs in the U.S. fail to improve the conditions of the large majority of patients they are prescribed to. More research needs to be done to determine why this is, but one reason is likely that the drug or dose dosage is not compatible with the patient's genetic makeup. We hope that the future of medicine allows for providers to offer pharmacogenetic guidance to their patients, and that if you are someday the patient in that situation, you have an understanding of how this could help improve your outcome.